Welcome to DNA and Candy and Books. I'm Miss Donna from the Little Lake Harbor branch.
And I'm Miss Cindy from the Tuckerton Library.
We are going to make a candy model of DNA. Plus, we have another DNA project and some good book recommendations for ages 9 to 13 or so. And plenty of information about DNA along the way.
Well, let's get started with this candy. To make this model of DNA, you will need 2 Twizzlers or red vines or whatever this brand is, and some gumdrops or the candy called dots, and you'll need some toothpicks. And you'll need to wash your hands first. Are your hands clean, Miss Donna?
Yes, they are.
Excellent. So, what you're going to do is start by putting about 8 gumdrops on some toothpicks. But not just any gumdrop. First, I want you to choose 4 colors and only use those. So, if you want to get started sorting your colors and putting some onto toothpicks, let me tell you a little bit about DNA. What is DNA? It is the instruction manual for your body. It tells your body how to make skin and bones and hair. And it also tells, that's how you put the gumdrop on the toothpick. Thank you. It also tells your body how to work, how to digest your food, how to heal if you cut yourself. So the instruction manual is written in a chemical molecule called DNA, which is short for deoxyribonucleic acid. You can see why people abbreviate it. Now, DNA has a unique and interesting shape, which is really why people like to make models of it. Also, figuring out the physical shape was key for humans to understand how DNA actually works. There is an interesting story about how the shape was discovered.
The shape of DNA was discovered by a female scientist, Rosalind Franklin. In this juvenile biography, Rosalind Franklin, discoverer, the book explores difficulties Rosalind faced as a female scientist in the early 1950s. On page 11, this is the famous photograph 51 that Rosalind Franklin and Raymond Gosselin discovered. In the early 1950s, research on DNA was made possible with new technologies back then. Rosalind Franklin discovered that DNA, when photographed with special equipment, looked like the letter X. When we hold up the candy model, you can see the letter X. If you want to learn more about how difficult it was for Rosalind to be a female scientist, you can read about it in this book.
So the shape of DNA, as Rosalind and others discovered it, is called a double helix. Helix means spiral, and double means the 2 sides spiral up together, like so. The sides of DNA are made of a phosphate molecule and a sugar molecule hooked together, and that is represented by the Twizzler. And the sides are connected like ladder rungs with the important part, the nucleotide bases. There are only 4 bases, and that is why you can only use 4 different colors of gumdrops. I'm really sorry, I'm afraid you're just going to have to eat the rest of them. So these 4 bases are like the alphabet of the DNA instruction manual. They are only 4 letters, and those letters are, Miss Donna?
A for adenine, T for thymine, G for guanine, and C for cytosine.
All right. We have A, T, C, and G. That's what everybody usually calls them. So Miss Donna has labeled hers, and I recommend that you do the same. But it doesn't really matter which color you call what. So I hope you've got yours sorted and you're putting your toothpicks on gumdrops. So the funny thing about these bases is that, like jigsaw pieces, puzzle pieces, you can't connect just any base to any base. A will only bond with T, and T will only bond with A. C and G bond together, but neither of them can bond with A and T. And that is actually a really important part of how DNA works. So At this point, if you've got your 8 or so toothpicks with one Twizzler on it, with one gumdrop on it, what I want you to do is put a second gumdrop, the right gumdrop, onto each toothpick that already has one. If you've got an A on one toothpick, then get a gumdrop out of your T bowl to put on the rest of the toothpick. And if you've got, say, a G gumdrop on a toothpick, take a gumdrop out of the C bowl to put on that toothpick. All right, while you're working on that, I want to tell you about some books, because if you're really interested in how DNA works, you might want to read this one, DNA Up Close by John Shea. It has a lot of good information, what I've said, and more besides, in a level that is right for 9 to 13-year-olds. And if you're a little more advanced in your reading, you might be interested in this one, called Genetics, Breaking the Code of Your DNA, by Carla Mooney. It has more information, along with plenty of little comic panels to help illustrate the points. So the shape of DNA is key to how it works to deliver the instructions to your body. You see, when it's time for the cell to do something, that's usually make a protein. So let's say you're a snake and you've just bitten somebody, and now you need to make some more venom so you're ready for the next time you need to bite somebody. Or let's say you're a human, you've met a virus, and you need to make an antibody to try to fight off that virus. So when that happens, the DNA uncoils itself and unlinks right along the center of the ladder rungs, the toothpicks. Those two bases unlink. And then something called RNA comes along and links itself onto that half of the DNA. The RNA is almost like DNA. It is also made up of 4 bases that can briefly bond to the DNA to make sure that it is an exact but opposite match to what's there. And then the RNA unzips itself from the DNA, which links back up and coils itself away. That RNA leaves the nucleus and goes out into the main part of the cell, where it finds a ribosome, which reads what that RNA says the DNA has on it, and uses that to create the proteins. So how does it do that? Okay, so the RNA is made of bases, these nucleotide bases represented by our gumdrops, and those are like letters. Well, the ribosome reads words. The ribosome reads those bases in groups of 3. Each group of 3 bases is called a codon, and it means a particular amino acid. Proteins are nothing but strings of amino acids. So the way the ribosome builds a protein is it reads the RNA. The first codon says start here, and then the next one, the ribosome reads the next 3 bases, that codon, and it knows what amino acid that codon tells it to get. There's a lot of amino acids just floating around. The ribosome grabs the right one, then it reads the next codon, the next 3 bases, grabs the amino acid represented by that, and hooks it onto the first one. Finds what the next one is, and gets that, reads the next codon, and gets that, and so on until it has reaches the end of the RNA, which says, you can stop now. And by this time, it will have written a whole sentence out of amino acids, which is, and we call that sentence a protein. So now the job is done, and the ribosome looks for a new piece of RNA so we can start the work all over again. Speaking of getting the job done, Miss Donna, it looks like you are finished with your DNA molecule.
Yes, and if you look at it, you can see the letter X.
Look at that.
Yummy, yummy.
Well, I think I might want to have one of those spare gumdrops now. I hope you've enjoyed making your candy DNA molecule. Up next, we are going to make a bracelet using some actual DNA coding sequences. I'm going to put the instructions for that up on the screen now so that you can download them if you want to. If you're not able to, don't worry, we have some other options for you. So, we'll be back in a minute. Okay, let's start our second craft. We are going to make a double-stranded DNA bracelet like the ones Miss Donna is modeling. It contains actual DNA code for a protein. One of them is a sequence from a gene that codes for an opsin protein, which allows a butterfly to navigate using ultraviolet light. And the other one is a sequence from a gene that codes for a toxin protein, which is a component of the Malayan spitting cobra's venom. And it blocks signals between the nerve and muscle cells of the cobra's prey, which paralyze it. Ooh, which one are you making, Miss Donna? Is it going to be the snake venom?
No, it will be the monarch butterfly.
Okay. Well, you at home can choose which one you're going to do. We are going to have this information available in the chat, and you may have just downloaded it too. Let's get started. For this project, you will need beads in 4 colors. Why 4 colors? I bet you know they represent the nucleotide bases, just like in the gumdrops. Your beads can be any size and color. You will also need some elastic that fits through the center of your beads. If you don't have elastic, you can use something else, such as maybe embroidery thread, like a friendship bracelet, but your knot will be different at the end of the bracelet. So, as we learned when we made the candy model, the alphabet of the DNA instruction manual is made of only 4 bases: adenine, thymine, cytosine, and guanine, which we abbreviate to A, C, T, and G. And as you remember, A and T will bond together, and C and G will bond together, but they cannot deal with the others. So that's a really important part of how DNA works and an important part of how you will put your bracelet together today. First, you need to decide what color bead is going to represent what base and label them. What are you using today, Miss Donna?
Yellow beads represent adenine. Crystal clear beads represent thymine. The green beads represent guanine. And the purple beads represent cytosine.
All right, let's get started with our double-stranded DNA bracelet. Now, we call it double-stranded because you're going to put beads on 2 different strings. The beads on one strand will represent the nucleotide bases attached to one side of the DNA double helix, like the first gumdrops you put on the toothpick. And the beads on the other string will represent the complementary bases, like the second gumdrops that you put on the toothpick, that attach to the other side of that double helix. Okay, we are going to tell you the bases that make up the first string for your butterfly or cobra toxin gene sequence. And you are going to have to use your scientific knowledge to figure out what to put on the second string. All right, so please start by cutting a piece of elastic string about 12 inches long. If you're going to be using something that doesn't stretch, you might want to give yourself a few extra inches because your knot is going to be different. And go ahead and make a second strand the same length. And then I want you to use a marker or something to color the end of one of those strings because it will be very difficult to keep track of which is which as you work. So it's really helpful to know. Miss Donna is using a green marker to just throw some color on the end of one of the strings. Now I want you to make a knot at the other end of that string. Oh, I see. Miss Donna is also using a glue stick on top of that marker for the ends of her strands. That's because we've learned that this string tends to unravel, and a little bit of glue stick will help hold it together as we string those beads. Okay, time for knots. At the other end of the string, I want you to tie a simple knot, an overhand knot, about 2 or 3 inches away from the end, and then do the same on one side of the other string. Once you've got that done, you're going to tie those 2 strings together. And the best way of doing that, as I have learned over the years, is to lay those 2 strings together and treat them as if they are just one piece of string. tie the same kind of simple overhand knot. Just make sure that it stays right where those first two knots are. Tie it nice and tight, so all three of those knots are all together, and then your beads will have no chance of slipping off. All right. While you're doing that, I want to tell you more DNA facts. Most DNA is the same across all animals and people. I share about 9/10 of my DNA with my cat. We all share about 40% of our DNA with a tiny fruit fly. I think that's crazy. 98% we share with chimpanzees. They're the animal that's closest to us. And the reason why we do why all this DNA is the same, it actually makes sense. Bones and skin are the same. Healing and digestion, it's the same across animals. So it makes sense that our DNA would be mostly the same. Okay. Let's see. Before we get started, Miss Donna, I bet you have a book to share with us.
Yes. In a juvenile book of science fiction, human and animal DNA is mixed. In Tentacle and Wing by Sarah Porter, there is a terrible science experiment that has gone wrong. Children in the story are born with both animal and human DNA. These children are called Kynes after chimera, for half animal, half human. In the story, Ada, the main character, is sent away from her family after a sneaky DNA test is taken of her, and she is discovered to be a Kyne. She is sent to live in quarantine with other children Kynes. While Ada is there, she tries to solve a mystery. If you want to discover who is in charge of the experiments and solve a mystery, check it out in this science fiction book.
All right. Of course, today we can't really mix animal and human DNA, but it makes for such interesting stories that other people have written it too. Older people might like to check out the Maximum Ride series by James Patterson. Okay, back to our bracelet. Each bead on your bracelet is going to represent one nucleotide base in a genetic sequence that makes up a protein. And here are the sequences for the two proteins. Now, you may have just downloaded this because we just put it up for you. We'll also put it in the chat so you can follow along the side because you're going to need to refer to that as you make your bracelet. Butterfly is at the top and snake venom is at the bottom. You get to choose what to do. So the instructions tell you which bases to put on your marked string, and then you are challenged to figure out what base to put on the other string because you know the drill. A and T bond together and C and G bond together. Now these colors may not match the beads that you're using. They don't match the bead color that Miss Donna is using, but that's okay. I recommend actually that you put your bowls together so that it's easy for you to connect them. Okay. Both DNA sequences, the snake venom and the butterfly, happen to start with A, the same base, A. So let's put an A bead on your marked string. And then what goes on the other string? What base connects with A?
T.
That's right, it's got to be T, so put a T bead on your other string. So, as you can see, whenever you put an A bead on one string, you'll put a T bead on the other string. When you put a T bead on your marked string, you'll have to put an A bead on the other string. And if you use a C bead on your marked string, you have to put a G bead on the other string. G bead on the marked string, guess what? C bead on the second string. Have you got your second bead on yet, Miss Donna?
Nope.
All right, what's your second bead?
T.
All right, she's putting a T bead on first string. And then what is she going to put on the second bead, on second string? It's got to be an A bead. So what is the third base going to be?
G.
A G. So she's going to pick up a green bead. And then she'll put what on her second string? A C. I think you probably understand how to do this now. So go ahead and keep working on your bracelets, and I will tell you more about genetics. All humans share 99.1% of our DNA. Miss Donna and I, and you, have almost all of it in common. But it's that 1% that's different from one human to another that's important, that makes us each unique. It changes our body shape, our hair, our personality. And because one person's DNA is a mixture of their parents' DNA, the people most similar to you are going to be your genetic mother and father, and their parents, and any other children that they may have created, brothers and sisters. And that is why people in the same family usually look like each other, but not completely. Miss Donna, is there anyone who shares your DNA absolutely completely?
No, I don't have an identical twin, and that's the only way someone else would. Scott Kelly has an identical twin, though.
Who's Scott Kelly?
Scott Kelly is an astronaut for NASA. They studied him when he lived in space, and his identical twin back on Earth. To learn more about Scott Kelly's life, you can read about it in this biography, Scott Kelly, Remarkable Space Resident.
Aside from identical twins, no one in the world shares the exact same DNA, and this can be really useful. For instance, in solving crimes. If we find some blood, which is one of the best sources of DNA, we can find out for sure whether it belongs to somebody or not. So if a criminal leaves some DNA behind, we can test it and know for sure whether they were there, even if they try to say they weren't. This kind of law enforcement work is part of a field called forensics. This might be a career you're interested in. If so, the library has a lot of different books you might be interested in reading. For instance, this one, Forensic Science, from the Jobs That Rock series, is a very interesting one that combines a story and some actual information on how scientists do their work, told in kind of a graphic novel format. It's really good. And another one that I would love to recommend, especially to older kids, is called Blood and DNA Evidence, Crime-Solving Science Experiments by Reynes. It has really good experiments and exercises that will actually teach you a lot of useful skills and knowledge that forensic scientists really use. And it explains exactly how they test to identify people by their DNA, which is something that I always kind of wondered about and found very interesting. So, Miss Donna, do you have a story to recommend?
Yes, forensic science is used in the third book of the Explorer Academy series, The Double Helix, a Novel by Trudy Truitt. The students scan fossils for DNA and use fancy gadgets to discover what remains were left on the fossil. If you look at page 20 and 21, you can see a hologram of a saber-toothed cat. The saber-toothed cat had left its DNA on the fossil. The students used forensics and advanced science to see it. If you want to find out what other ways the students use DNA to solve mysteries, you can read it in this fun fiction book.
All of our books are written in a book list, which is available as part of the download. So we have only been able to use DNA a little bit longer than you've been alive. But it's a really good tool. In this book by National Geographic called Genetics from DNA to Designer Dogs, they talk about all kinds of different ways that DNA is used in many different fields, such as history, medicine, fighting pollution, and saving wildlife. And it uses a lot of examples of actual people and real situations. So how is that bracelet coming, Miss Donna?
Well, it's a little difficult, but we're coming along.
Oh, I can see how the yellows and the whites are always opposite each other, and the green and purple are opposite each other.
Yes.
All right, well, while you continue to work, let's talk about viruses and coronavirus and how a virus works. So first, our bodies are made of many, many, many small cells, which are too small to see with your actual eyes. You need a microscope. When you get sick, it's probably caused by either a bacteria or a virus. Now, a bacteria is way, way smaller than a cell, but it is a living thing, which means that it grows, it eats, it reproduces, and it moves. So a virus is even smaller than a bacteria. You can't even use a regular microscope to see it. And it is not alive. It never eats. It never grows. It can only move if it's attached to dust or a water droplet, like in your sneeze. And it doesn't even reproduce. It's just a tiny scrap of DNA or RNA and a delivery mechanism. It can't do anything except trick us into reproducing it. So let's talk about that. When a virus, and not just coronavirus, flu virus, cold virus, there's a whole lot of other viruses too. When it meets a cell that it can work with, that delivery mechanism that I mentioned just gets the main part of the virus inside the cell. And then that scrap of RNA takes over the ribosomes in the cell. Remember how they're supposed to be used to make proteins? Well, the virus crowds out that important work of the cell, and it just makes the ribosome do nothing but make more and more and more copies of that virus. When it has made hundreds of copies, it bursts the cell, which kills the cell and doesn't do you any good. And then each of those hundreds of copies make their way through the body in search of more cells to infect. They get inside and make hundreds of copies of themselves in those cells and kill those cells, and then all of those thousands of viruses go hunting for a new cell. But all of this virus making doesn't actually do the virus any good. It's not alive, and it certainly is bad for us. So it's a good thing that our bodies have a lot of ways of trying to shut viruses down before they can get in our cells and kill them. It's pretty good at doing this. Lots of times it works perfectly well and we don't get sick even though we've been exposed to something. But this is why it takes a while for you to get sick, if you do, once you've been exposed. Because the first virus gets inside you and it takes a long time for it to go through enough of these cycles that it kills enough cells that you start to notice symptoms like a fever or a cough. So this time period between when the first virus gets inside and when you notice symptoms is called an incubation period. With coronavirus, it's 2 to 14 days. So if we think we've been exposed, we quarantine for 14 days so that we can figure out whether our bodies have stopped any viruses that may have gotten into us. If you only inhale a small number of viruses, it's easier for a body to stop them all. But if you take in a whole lot, like say you're talking to somebody who's sick for a long period of time, then they can infect a lot of cells faster and make more viruses so fast that your body has a much harder time stopping them all. But I think it's interesting that the virus is really nothing but A, C, T, and G bases, just like you and me and your bracelet. So, Miss Donna, have you finished your bracelet?
At this point, I say we'll stop.
All right. I think what we're going to do is pause our video, make more of the bracelet,
Sure.
and then we'll pick it back up to show you how to finish yours. Okay, Miss Donna, have you finished your bracelet?
Yes. Let's show them how to finish up.
Okay. Ooh, that's looking nice. So, when you've got all of your beads, tie another knot at the end of each thread. Hold the strand up so all the beads kind of fall down to the bottom, And then tie a single overhand knot, like you did before, about a centimeter, half an inch, a bead's width or two away from where the beads stop. And be careful when you set it down because the beads might still be able to slip off. We're not done yet. And then tie another knot just like that one in the other strand. And then hold those two strands together as if they are a single strand and tie those knots together with a single overhand knot, just like you did at the very beginning. And then the beads will be secure and will not be able to fall off. All right, I think Miss Donna's ready to tie those two knots together. She's laying the two strings together with holding the knots together, wrapping that around, pushing both of them through, and tying the knot. Oh, it didn't work. It usually happens, doesn't it? Second time around, tying those two knots together, and then making it nice and snug right where those two knots connect. Do you have all three knots in the same place, Miss Donna?
Yeah.
Okay. Okay, so now we have all of our beads on our two strands. We have each strand knotted and the two strands knotted together at each end. To finish the bracelet, we have to loop it back around and lay all four strands together and make a single overhand knot right where all of those other knots are to finally make our bracelet into a loop. Go ahead and work on that if you can, Miss Donna. If you are not using elastic string, then what I recommend is that you look for a YouTube tutorial on doing a bracelet sliding knot. There are quite a few tutorials out there, and that will allow you to put the bracelet on and off without having it slip free. So once you've got that done, put it on and admire it. You'll want to cut the loose ends off of your bracelet, but I recommend putting a drop of glue on that so that they don't fray and lose it. All done. Oh, look at that. I admire that a lot. And we can see that the bases, oh, I'm not social distancing, that the bases are paired up nicely as it goes along. Nice work, Miss Donna. Excellent.
Thank you. Thanks so much for attending our program. We hope you learned something about DNA and enjoyed making our crafts. The books we recommended are included on the book list that's available for download. And we want to thank Little Bins for Little Hands and YourGenome.org for the craft ideas. Happy reading!
Happy reading.
Support public libraries. Like, share, and subscribe for more great videos.